Hi students, welcome to the last part of the Chapter 12 lecture. So we've gotten through discussing the action potential itself as well as the graded or local potential, which you can think of as a blip in a graph like this. So the action potential itself is illustrated here. Remember that our resting membrane potential for a neuron is about -70 millivolts. If it's a local or graded potential, you're just going to see this slight disturbance. You're going to see a slight depolarization. And if it remains a local potential, it's just going to fall back down to resting. But if the stimulus is strong enough sodium gates are open, causing that true action potential to occur, that threshold value of about -55 millivolts is kind of the magic number here. So we have this quick up and down shift here. We have depolarization. That's a really sloppy arrow. We have depolarization and then repolarization occurring. And there is a point in time during this firing of an action potential in which you cannot get that same neuron to fire again. So during an action potential itself, and for just a few milliseconds after, it's going to be either difficult or impossible to stimulate that region of the neuron to fire again. And remember that a neuron itself can have multiple action potentials occurring basically simultaneously, but we're talking about a very localized area. So a particular area of an axon has this what's known as an absolute and a relative refractory period. So the absolute refractory period is essentially the period of time during which no strength of stimulus can cause that region of the neuron to fire an action potential again. And you'll notice that this is the area basically in the immediate vicinity of when the action potential is occurring itself. So essentially what this means is you can't have overlapping action potentials in the same region of the same neuron at the same time. And then moving further away from the point past which we've gone back hyperpolarized even, we have this relative refractory period. And that's the period of time that follows the main part of your action potential, during which only a very strong stimulus is going to be able to stimulate that neuron to fire again. So again, we have our absolute and our relative refractory period. Our absolute refractory period, you're not going to have any possibility of stimulating that region of the neuron to fire again. Again, you basically can't have overlapping action potentials occurring in this same region of the neuron. This lasts as long as sodium gates are open, and then it's inactivated, and then you shift into this relative refractory period. So this is a period of time during which it's just very difficult to stimulate that neuron to fire again. So only an especially strong stimulus is going to trigger a new action potential. During this period of time, potassium gates are still open, and any effect of the incoming sodium is opposed by the outgoing potassium. So generally this lasts until hyperpolarization ends. And remember that hyperpolarization is the period of time during which you are still working to get that resting membrane potential reestablished. So there's a brief period of time during which the cell is actually hyperpolarized. The membrane is more negative than normal. And then you have your sodium and potassium pump as well as your sodium and potassium channels basically reestablish that resting state. So it is important to note here that only a small patch of a neuron's membrane is going to be refractory at any one time, and other parts of the cell can still be stimulated. All right, let's talk about conduction speed of nerve fibers. So conduction speed, basically another term for this, is conduction velocity. So how fast is an action potential actually occurring? How fast is it traveling down the axon? So it's the speed at which a nerve signal is traveling along the surface of the nerve fiber. And that's going to depend on two factors. You have your diameter of the fiber, so the larger fibers have more of a surface area, and therefore they're going to be conducting signals more rapidly. And then secondary to that is your presence or absence of myelin. So remember that myelin is an insulative wrapping that's formed by, in the CNS or central nervous system, that myelin sheath is going to be formed by oligodendrocytes. So that's a specific type of glial cell that forms that insulative wrapping. And then in the PNS or peripheral nervous system, we have our Schwann cells. So functionally, they basically do the same thing. Anatomically, they're a bit different from one another, but they both contribute to this fast conduction velocity. So let's compare the conduction speeds here. Remember that the larger the diameter of the fiber and the more myelination that is found there, the faster the speed at which you can conduct an action potential. So a small unmyelinated fiber is going to conduct an action potential in this range of 0.5 to 2 meters per second. A small myelinated fiber is going to conduct at 3 to 15 meters per second, and then your large myelinated fibers are going to conduct it up to 120 meters per second. So our slow signals are generally going to be confined to signals that do not rely on the fastest possible action. So generally those signals that are sent to the gastrointestinal tract, where speed is less of an issue, those would be governed by those smaller and unmyelinated fibers, whereas faster signals are going to be sent to skeletal muscles, where speed improves balance and also coordinates body movement. So you're going to see reflexes and signals that are sent to skeletal muscles as being your fastest conduction velocity. There are a couple of different types of conduction depending on whether or not you have myelination in the fiber. And remember, just as a reminder here, sometimes it is difficult to remember, what exactly are we talking about with a fiber? What is a fiber? Remember that we saw that term with muscle fibers. That was just another term for a muscle cell. And in the case of a fiber in the nervous system, the fiber is just the axon. So remember, stylized neuron here. So this is kind of a generic neuron here. You have your cell body, the soma, and then this entire length, this tail-like structure, is going to be your axon. And because it is long and thread-like, it's referred to as a fiber. Excuse me. And then remember that a nerve is just going to be bundles of fibers. So to have a nerve, say your cranial nerves or your spinal nerves or your vagus nerve or phrenic nerve, any of these major nerves, they're going to be bundles of many of these axons or fibers put together. So our unmyelinated fibers are going to have voltage-gated channels along their entire length, and an action potential at the trigger zone, which is the region between the soma and the axon. That trigger zone, that's where enough sodium channels exist where you can actually have propagation of an action potential. Your action potential at the trigger zone is essentially going to cause sodium to enter the axon and diffuse into adjacent regions. So that depolarization then excites more voltage-gated channels, and those opening voltage-gated ion channels are going to result in new action potentials being propagated, which then allows sodium diffusion to excite the membrane immediately distal to that. So it's basically this chain reaction that just flows unidirectionally from the trigger zone all the way to the end of the terminal, or the end of the axon there. So it's a chain reaction that continues until the nerve signal reaches the end of the axon. So you can kind of think of it as if it's a chain of falling dominoes. So here we have a depiction of a typical neuron. We have our dendrites, which are often going to be in contact with other neurons, or there might be free nerve endings here. So they might be the first chain in your typical 3-chain pattern of sensory, interneuron, and motor neuron. But you're going to have your stimulus that ultimately causes sodium gates to open within that trigger zone. And then you're going to have this chain reaction where you have action potentials occurring along the length of this axon all the way to the end where you have your synaptic vesicles. So what this is showing is basically just a depiction of an axon here, a section of an axon. And you'll see your more positive ion concentration outside the axon here, your more negative concentration inside, except it's basically switched during this action potential itself. So the action potential in progress is illustrated in this reddish color here. Your refractory membrane is going to be immediately behind that where the action potential just was, and then ahead of that is going to be your membrane that is still excitable. So the refractory membrane helps to ensure that the action potential is only flowing unidirectionally, and it's eventually going to make its way to the axon terminal there. Now let's look at myelinated fibers and the difference between how the signal is conducted. So our myelinated fibers, rather than just being this bare axon, we have these oligodendrocytes and Schwann cells that form these insulative layers around sections of your axon. So a myelinated fiber is going to conduct signals with what's known as saltatory conduction. Saltatory means leaping. And it's called this because the signal appears to jump from node to node. Now that's not actually what's happening, but the signal travels so quickly within those myelinated sections that it looks as if the signal is jumping from node to node. So the nodes of Ranvier are going to be your bare sections of axon, whereas your internodes are going to be your myelin-covered areas. And the nodes of Ranvier contain many of these voltage-gated ion channels, which have the potential to open up, allow that influx of sodium, and then allow for an action potential to occur via that depolarization. The myelin-covered internodes contain few of those voltage-gated ion channels. So what happens is when sodium enters the cell at a node, its electrical field is going to repel positive ions inside the cell, because remember that like repels like. So if you have an influx of positively charged sodium ions, they are going to repel positively charged ions inside that cell, and they're basically going to flow again in the direction of the axon terminal. So as those positive ions are moving away, being repelled by that influx of sodium, you do kind of have a chain reaction again, where you have positive charge repelling positive charged ions, and you have this transfer of energy down the axon. So that's how we get the conduction of the signal in myelinated axons. So how the myelin speeds up that conduction is essentially by minimizing leakage of sodium out of the cell and further separating inner positive ions from attraction of negative ions outside the cell. So what we essentially see with a myelinated axon, if we have our nodes of Ranvier being the bare sections where you can have this influx of sodium, and then you have your myelinated sections, and then again we have a node and then an internode being the myelinated section. What actually happens with the myelin sheath is it helps to insulate the ions inside the axon from the ions that are in the ECF, the extracellular fluid. Remember that opposites attract. So if there are lots of positively charged ions within this cell environment due to that influx of sodium ions, they are going to be attracted to any negatively charged ions that are in the ECF. So they're going to want to basically bump up against your membrane because they are attracted to the ions outside the cell. What the myelin sheath really does is it further separates those oppositely charged ions. So in the nodes, you have more of an attraction for the ions that are inside the cell. They're bumping up against the inner part of the membrane because they are attracted to the oppositely charged ions outside the cell. But once those ions are in the part of the axon that has this myelin sheath around it, the myelin basically acts as an insulator. So those charged ions are going to flow more quickly through these insulated areas because there's just more of a separation between the ICF and the ECF. So there's less of an opportunity for them to essentially be pulled away from the middle of the axon by their oppositely charged counterparts outside the cell. So the myelin is going to speed up that conduction by minimizing leakage of sodium out of the cell for one, and then also further separating your inner positive ions from attraction of your negative ions outside the cell. Eventually, we do see the signal strength of our action potential starting to fade within the internode, within the myelin sheath. But when the signal reaches the next node of Ranvier, then it's going to be still strong enough to open more voltage-gated ion channels. And again, you're going to have this influx of sodium, and then a new action potential is going to be conducted down the length of that axon. So let's look at a nicer illustration of this. Here we have our cell body, our soma, and we have our dendrites over here. Here's our axon. And if we're looking at kind of a close-up view of this, we have our myelin sheath. We actually know that these are Schwann cells, so we're in the PNS with this illustration because of the fact that you have just a single cell that's wrapped around this nerve fiber. So we have an influx of sodium. Sodium is going to flow in at the node of Ranvier, generating an action potential. We have the positive charge that flows rapidly along the axon and depolarizes the membrane, and the signal is eventually going to grow weaker the further it gets away from this node, but you're still going to have depolarization of the membrane occurring. So the depolarization at the next node is going to be opening more sodium channels, and that triggers a new action potential. So with the saltatory conduction, because you have the conduction velocity through these internodes being so quick, it essentially looks like the action potential is leaping. It's going so fast that it looks like it's just skipping over these myelinated sections altogether, and hence the term saltatory conduction. So the takeaway message from this is, of course, that your signal conduction is going to be much faster in your myelinated fibers than it is in your unmyelinated fibers. And we still have this refractory period that we have to keep in mind. Refractory membrane is going to be where the action potential just was. Action potential in progress is illustrated in red here. And then the membrane that is excitable, that can respond and become depolarized here, is in green.